Well, good afternoon. My name is Tim Schroeder. I'm CEO and founder of CTI, a clinical research organization that specializes in regenerative medicine. I'm joined here today by a illustrious group of individuals who will give their own introductions, but our topic today is immuno-oncology, and we're going to talk specifically about commercialization issues, obstacles, and as these first products are approaching that particular stage, there's some really important things that really need to be considered. And so I'll start, Oz, if you'd like to introduce yourself and a bit about the company.
Sure. Hi, good afternoon, everybody. My name's Azam. I'm the president and CEO of Tmunity Therapeutics. It's a startup out of Philadelphia, spin out of the Penn ecosystem. Currently, we are funded by Penn and Lilly Asia Ventures that was seeded last year, and we're currently raising a Series A funding and moving assets into the clinic in the oncology space, both CARs and TCRs, and also working in the HIV and autoimmune disease space.
Thanks.
Great. My name is David Epstein. I'm executive partner at Flagship Pioneering, and I'm also chairman of a company called Rubius Therapeutics. I think that's why I'm here today. Rubius is a company that has licensed technology to go from stem cells to fully functional red blood cells, and then we genetically engineer those cells to create allogeneic platform to treat a variety of different diseases.
Thanks, David.
Hello, Aaron Foster, Vice President of Product Discovery at Bellicum Pharmaceuticals. Bellicum is a company that's been around for a little while now, located in Houston, Texas, sort of a center for cell and gene therapy. Bellicum focuses on switch technology as our core platform. We have now several different types of switches. The most well-known is the ICAS-Base9 suicide gene, which we employ in a haploidentical stem cell transplant to mitigate against graft versus host disease in those transplants. The second part of it is really an on switch, which utilizes some novel co-stimulatory molecules, but this turns on cells to keep them alive and let them proliferate a bit further. And we're employing this in CAR T cells and alpha-beta TCR targeted cells. We have an ongoing clinical trial. Our most advanced BPX-501 is focused around haploidentical transplants with a T cell engineered with ICAS-Base9 as an add-back therapy for both non-malignant patients, pediatrics, in primary immune deficiencies and other types of blood disorders, as well as in malignant cases for the treatment of, say, high-risk acute myeloid leukemia and other types of diseases.
Thanks. I really want to thank Aaron because he's pinch hitting for a colleague on relatively short notice, so we appreciate you joining us.
No problem. Appreciate it.
And Jeff, back by popular demand.
Yeah. I think I'm the least qualified person to be up on the stage right now, but Bluebird Bio, I'm the CFO of Bluebird Bio, and as it relates to immuno-oncology, we have a CAR T program in the clinic. We disclose data on that program. It's partnered with an amazing partner, Celgene. It's targeting BCMA for multiple myeloma. We expect to have more data coming out at ASCO on that program. That's our CAR T anchor. We're also targeting TCRs as well. We've got two relationships there that are noteworthy. One is with Kite, and the other one is through a relationship we have with Metagene to identify and develop other targets, TCR targets. So we're operating both CAR T and TCRs, and we also have a pretty robust tool set behind that is relevant for certainly this field, including a gene editing platform called Megatals that is really starting to emerge as a pretty exciting gene editing platform. And then we also had some other tools and technologies like switches that we're deploying at least preclinically now and hoping to move to the clinic.
Thank you very much. We are going to move through basically three big topics, starting with the CAR Ts. And one thing I'd like to do is encourage the audience with any questions. Just make sure you raise your hand for a mic so that we can hear it. We've got about 40 minutes here, though, but I think we'll jump right in, maybe putting the proverbial cart after the horse. But what do we think these labels are going to look like now that we're getting so close to potential approvals? Oz, you want to? Well,
So I think, look, it's a really exciting time for the CD19 space. I think validation of that target is now happening. You've seen the news with both Kite and Novartis having filed successfully. Doesn't mean that we are a slam dunk yet with approvals, but clearly both companies I know have been working extremely closely with regulators. And I think the body of evidence for certainly the indications that are being pursued are very targeted in the case of Novartis with pediatric cancers being focused on with acute lymphoblastic leukemia, diffuse large B-cell lymphoma for Kite. I think you're going to see, my prediction is good attractive labels in terms of those populations. There'll be clear guidance on patient selection. There will clearly be a highlight for long-term follow-up. This is first-in-class transformative therapies. There's a lot we don't know yet that the real world is going to establish. I think over the past 5 years, the body of evidence around benefit risk has been pretty well established. We understand now cytokine release and how to manage that. We understand B-cell aplasia and other factors, but obviously of late we've also seen issues around SAEs that maybe weren't anticipated, such as cerebral edema that set certain programs back in the landscape. But overall, I feel really bullish. I think this is going to be great hope for patients who have very few other options. I think it's going to be very competitive towards versus stem cell transplant. And let's wait and see. But the real world approach over the next 5 years and how that data comes out, how these patients really perform in a real world setting, I think is going to be key. But I believe they're going to be good labels.
I think it was really well said. But if Mike's on, if I could add to it, unlike a traditional antibody or small molecule, the battle is not going to be won or lost with the label.
Yeah.
There's a lot of other things. For example, how you get the patient sample, get it to the factory, process it, get it back to the patient. How many samples are lost on the way, how the patients are treated, how quickly things get turned on. All these things are going to make a bigger difference, I think, initially than the label itself. And companies are building that capability. It's hard to get clear visibility on who really has it worked out at this point.
Yeah, I guess I would add, from our perspective, our most advanced program is not a CAR, but it's a polyclonal T-cell population. But some of the same problems apply. I think that what's been really interesting to watch, Novartis in particular, and Kite for that matter, is the educational process that's getting involved with clinicians, trying to take these things, which are very novel and function very differently than any other types of therapies that we've given patients, and try to educate physicians on how to use them. And that seems to be working, which is quite interesting. And we're going through some of the same growing pains. The second point really is about logistics and manufacturing, where we have these internal portfolio discussions about what indications to get into, and people say, oh, well, that's 150,000 patients. I can't imagine making that many cells right now. So, the ramp up for getting this, making enough cells for patients when it's approved is going to be a very important problem to solve.
Jeff,
I don't know if I have anything to add other than I completely agree on the manufacturing side that that's going to be a critical component, and frankly, all of us are anxiously waiting to see how well out the gates, in particular, Kite and Novartis do in terms of that scalability and not stumbling along the way. So I think it's great points.
The challenge, said very simply, is to make money. You have to get to scale because you have to have a lot of patients being treated. But at the beginning, you're probably not making those investments, or perhaps you're not making those sufficient investments because you don't have anything to sell and any profits to invest in. So, and if it really catches on, you may find one or more companies that are behind the curve in terms of their ability to manufacture.
But I think if you link the label to manufacturing, probably the interesting thing to look out for is, in my opinion, adaptive manufacturing. So not all patients are made the same, particularly in the heme malignancy space, different disease populations, different tumor burden. And so I know that the companies have focused on this, and it's going to be interesting to see how regulators look at this, because it's not a fixed process. The product is the process.
Yep.
Yeah. Yep. So that's going to be probably the rub in the label, I think, and I'm curious to see how that's going to pan out.
Before we move off the label, we've got some very experienced pharma executives here, where historically it's been get a label and expand over time. Do we think that's a strategy here? And if so, are some of the lead companies already in a position to be doing that shortly after launch, or do we think that's, to your point, David, they're so focused on infrastructure that maybe that isn't front and center?
It depends how you define expand. I mean, typically in our industry, we've said indication 1, 2, 3, 4. And that may be the case, for example, with the anti-CD19s, but then maybe we can make them better. Or maybe there's another target, or maybe it needs to be human or not. You start getting into multitude of possibilities. I think there's going to be more of an evolution and course corrections along the way than just the sort of linear thing that we're used to in our industry.
And going from sort of refractory relapsing third line populations, I mean, that's a finite population moving up. I mean, that's a whole sequence there, and then moving out from, bone marrow transplant centers, for example, in the case of CARs and hematological malignancies, moving that itself into, a secondary setting or primary setting. That's a whole minefield in itself. So within the CD19 space, there's huge possibilities that still need to be addressed before you even start looking at BCMA, myeloma, and other targets. So I think there's huge room to grow.
I think playing off of that, I think one of the keys to that, clearly tying back to your real world evidence, is actually how the safety profile plays out over time in a larger population. I think ultimately, obviously the efficacy has to hold, but that's going to dictate ultimately how comfortable we all are in moving upstream into patient populations beyond the relapse refractory.
Jeff, how are you and your partners thinking of this what does this commercial model look like at the launch, and how does that evolve over time if it does?
So we're reasonably far from that, given that we're in a phase 1/2, but we do have a fair amount of experience now leveraging what we're doing in thalassemia and ALD, at least in terms of thinking about it and engaging with hospitals, in particular in Europe for our thalassemia program, as we started to build some commercial infrastructure there. It's going to be a COE type model where you have centers of excellence around which you center the patient, and then you build the support mechanisms around that. It's complex. It is very patient centric, and it's hospital by hospital initially. It is really going hospital by hospital and trying to understand, probably at the CFO or above level, to engage in a dialogue on setting this up the right way so that we don't stumble. Because one stumble here or there is really going to hurt not just that organization, but it'll hurt the field a bit. And so I think we all have to be very thoughtful and diligent about how we go about launching these products. I can't fully speak for the CAR-T and oncology world. It's actually a world beyond us, as we haven't been focused on oncology commercialization and clearly haven't been there, but some of the other folks here may have more insightful comments.
I mean, from a commercialization perspective, I mean, the obvious low-hanging fruit is going to be the clinical trial sites that have been actually the champions and advocates, right? And that's where I think the initial focus is going to be with the companies. If you get that right, you make a first mover advantage, and to David's point, the chain of custody, the chain of identity issues are locked. You've locked in your reimbursement mechanisms. You've really trained the physicians, and in many cases handheld them, right, in the early days of CAR-T therapies. It's going to be, in my opinion, a slow, steady build. This is not going to be a rapid adoption to start with, because these are powerful. T cells are powerful, right? They replicate and they grow and they expand, and we've seen the consequences of that in the early days of the CAR field. So I think a slow, steady build is going to be in order here, starting with the clinical trial sites.
And Oz, do you see the international expansion following a similar path?
Sure. I mean, it depends on, again, the geography, right? Depends on the business model in those countries, right? I mean, even just looking at Europe, it's not homogeneous, it's heterogeneous from a healthcare system, right? But I think the same principles are going to be involved. Obviously, more challenging discussions about reimbursement. That's going to be an interesting nuance, and we had an interesting panel discussion on that earlier on. But I think there are going to be a lot of principles that are going to apply across the globe. And then, obviously, ultimately scaling these therapies, and the more patients you can get through, the more efficient your cost of goods reduction are, the more volume you're going to get, and the better returns as well from a company standpoint.
I think one of the sorry for jumping in, but one of the cool things here about this is we really are setting up a completely new way of treating patients, and clearly the clinical data are driving this, and it's quite remarkable, some of the oncology data that are coming out. It's quite exciting. But this group of this particular Kite and Novartis initially, and then others to follow, hopefully us as well, are really creating a next wave of therapeutic intervention that is unbelievably exciting. You fast forward 10 years from now, I think this will be a standard. We might stumble in the beginning, and probably should expect that we'll stumble a little bit, but this is going to be a new way of treating patients across many diseases. How broad that is, I think that's still a question, but it's going to happen, and it's pretty amazing to be a part of it.
Do you think, Jeff, have we set the bar too high, the expectations in the investment community and the partners that, as you said, this is so transformative that if we fall short, we end up with a repeat of a Dendreon or something?
Yeah, no. If you look at the clinical data that's emerging, that's what's going to drive this for sure. And I can't imagine, given the clinical data you're seeing across all these indications, that this is going to go away anytime soon. In fact, it's quite the opposite. It's going to, I think, accelerate.
I agree. And I think for investors, it's a question of whether you're a short-term investor that's managing by the quarter or you have a longer-term horizon. Because clearly when these products launch, there's not going to be profits right away. And you always run that risk that as soon as you are a commercial company and then you have to generate EPS, that you disappoint or you do the wrong things to manage the short-term EPS. But I agree fully with your perspective on the medium to long-term about what the opportunity is.
Any other comments?
Well, I'm as excited as anybody on this panel about the potential. I mean, I think you look back at CAR T-cell therapy in general, and it's a truly remarkable technology. And I was in academia for most of my career, and we always believed in this. And the pushback we always got from people that wanted to have them invest in our technology was that it just wasn't going to be feasible. And I think the turning point, of course, is clinical data. When the CD19 studies start to come out, you look at that and you say, that's awesome, right? And now it's really the community and larger companies trying to figure out how to make it work, and there'll be a whole bunch of other people on the side of trying to figure out how to get paid for it. But given how well it works, there's going to be a way. And I think it's, in my mind, just taking that initial enthusiasm and pushing it into other indications, solid tumors in particular, and how do you generate technologies to start to get those to work, to expand the field from CD19 to myeloma and other types of diseases.
What do you think? I mean, for most new agents, you learn a certain amount in a clinical study. The protocol's defined, the doses are defined, the monitoring is extremely rigid. Suddenly it gets out into the real world, and then we find out how to really use a drug. In this case, how do you see that evolving? What don't we know, do we think, at this point in time?
None of these drugs are the same. That's the complicated nature of it. Not only are they individualized at this point, but the constructs are all different. The methodologies that people use to condition their patients are generally different. The patient selection can be different. And the quality and expertise of the physicians treating the patients can be different as well. So, I mean, if you just go back to the molecular biology of what we're doing and the cell types that we're trying to engineer, even from trial to trial at the same company or same university, you get completely different products. To me, it's really, you look back and these are small clinical studies that are basically where you're doing your experiments. You can't model these things in animals. There's not really any good predictive assays to understand whether they work, so you have to do these clinical studies like people have done in CD19 and see what happens and iterate from there. There's almost no other way to do it.
Do you think there's even more unknown for these therapies than a traditional biologic launching?
I think yes, simply because we learned a lot over decades about how drugs distribute or how antibodies react and what you can expect in terms of immunogenicity and these surprise things that happen. Here it's the beginning of a new field, so almost by definition there's going to be more surprises.
I mean, we're going to understand better, right, how these therapies really are persistent and durable at the end of the day. So, the CAR T field, I mean, we kind of think it's been the past 5 years as a renaissance. If you look at the practitioners, this is a field that's been evolving over 25 years, 30 years, right, with the early HIV experience and then evolving through, right? And there's still so much we don't know from the safety side, we still have to understand off-target effects, conditioning. We talked a little bit about that. I think that's going to be a huge driver of how patients are managed and the nuances in how physicians individually tinker with their regimens. But at the end of the day, actually, you're going to still be seeing patients who maybe do well and then relapse.
Yep.
You're going to see escape. Cancer's a formidable foe, right? I mean, the mutations will be there. You're going to get CD19 relapse, CD19 negative disease, and that's just the CD19 space. So huge opportunity still to do more really good discovery and development work to optimize for the future. And I think, this is the beginning of, a long journey still ahead of us. And I echo David's point that, it's going to be, you cannot expect miracles from a commercial return overnight here. There is an ecosystem that needs to be developed with the therapies coming out with these labels. But I do believe that the next decade is going to be pretty formidable for these patients who have frankly very little other choices.
I mean, for those who heard the earlier discussions about the payers and some of the challenges that we're seeing or anticipating seeing there, any suggestions at this point in time based on where we are with these programs in development?
Having come out of negotiated with payers most recently for a small molecule and for an antibody, I wish the people that are doing these first launches lots of luck, because they're going to set the path for the rest of us who are going to be coming later, hopefully with even better things. But seriously, I'm sure you discussed it earlier. What's the price level? Do you guarantee an outcome? Do you redo the procedure if the cells don't expand? All these questions will have to be addressed, and companies will decide to differentiate themselves in part by that commercial offering that goes well beyond just what I'm delivering to you.
Mm-hmm. Any questions from the audience before we move on from the CAR-Ts?
Natasha Rogosian. So in terms of your comments on manufacturing, can you comment on the importance of automation in your manufacturing and also when that decision needs to be made in the clinical trial timeline?
I'll kick it off and I'd welcome others' thoughts.
I think you have the most manufacturing experience on the stage, don't you, at the moment?
Possibly. So I think, automation is going to be really key, but it's one part of the puzzle, right? So, the CART process as they are now, they're a pretty fixed process, right, for generation one, right? And I don't think, there's a misnomer that people think this end-to-end automation is going to occur and it's going to be a cell therapy in a box. I just don't think that's going to happen overnight. But I think there are distinct steps to the process that those who have been in the field for many years have started to crack. So you can easily see, a future where cell growth, for example, can be really automated. There are established methods in finish and fill now for cell therapies that you can see a real driver of efficiency and reduction of cost of goods. So I think when you break it down, there are individual elements that lend to automation rather easily. Then you have others that actually, the companies are created around this. I mean, we have friends here that have such companies that look at the end-to-end automation. I think that's a more ambitious goal, but it's doable. And right now we're discussing autologous therapies. If you start to put on an allogeneic hat, which is where likely the field will move and the future will evolve, that's going to require, really, really good automation. But I think the key is your question of when do you do this? The earlier you do it, the better. So when you get translational medicine and manufacturing science meeting together and you put reverse correlation of patient findings in clinical trials, that's when you really know, is the automation going to work? So that's my opinion.
I was just going to add to that. I think that the process sciences part of the equation is very important. It's not just automation, and you can see that Bluebird and others are doing this. Where, what do you need to do to make your cells as fit as possible? And unfortunately, we don't really know what that formula is, or what cell type it is, or what composition of the cells need to be. And as we start to discover and again see clinical correlates with how we're producing the cells, or what cytokines, or what transduction methods, all these kind of very complicated things that affect the cell biology of the T cell. As those start to become understood, you can see how the process might get modified, even shortened, if you will. So, we and I think others have kind of bootstrapped our approach into the field to get it going, to get patients. It's acceptable and it works, but it's surely not the end. And there's going to be a long period of development that we are engineering these cells in a better way.
I think one of the interesting things is to play off that I think we're all going to watch play out is making these process changes over time. So getting back to a question you asked, do we know exactly what's resulting in either the efficacy or the safety profile or any of the other aspects of the therapy's kind of profile? I'm not 100% sure that we know exactly what in the process from collection to expansion to scaling up the vector, whether we make changes, how that's going to impact clinical outcome, because there are multiple components to this, unlike a small molecule therapy where you end up with a product or a biologic. What's going to be interesting is to see companies trying to figure out when to both automate, because that may change things, or when to implement process changes from version 1.0 or 2.0, whatever version we're on, and how to do that carefully so that you're not jeopardizing clinical outcomes. That's a fascinating conversation I'm sure is happening at every company that's thinking about this.
It's going to be interesting to see how the labels project
Yeah.
the manufacturing CMC section, right? Because it's not going to be boilerplate, right? It's going to be different as you know, you look at your different patient populations. So I'm looking forward to reading those labels.
Well, and you wonder how that conflict plays out between the, you know, we want to improve the process and reduce our cost and get more efficient at it, and at the same time, are we willing to take the risk that it may have a negative impact on clinical outcome, or minimally, we have to redo a bunch of studies and show that we're not changing something and all the time and cost associated with that.
Well said.
Yeah, or a positive impact. I mean, certainly some of these things could positively impact the cells that ultimately end back in the patient.
Yeah, and it's not just about, again, you know, we all think about this because autologous cell therapy is very expensive to make, and there's going to have to be some sort of automation or some sort of change to the cost of goods to make it a bit more profitable. I think we're all looking at that. You know, from our perspective, I think we have a number of different programs that are occurring sort of simultaneously, early clinical phase trials that are more advanced ones, and you know, so it gives us an opportunity to sort of assess some of these different process characteristics, if you will, on different stages of our T-cell therapies, and I think other people are obviously doing that as well.
Any other questions from the audience at this point? If not, we were going to move to solid tumors. The panel had some particular views there about what will it likely take to move the needle in a positive direction in the solid tumor area to at least move it forward similarly to hematological malignancies. Anybody want to jump in with the first thoughts or opinions?
Sure, I'll weigh in. You know, we've spent a lot of time looking at this. I think, like many in the field have. I think generally we feel that, you know, T cells, you know, lack the sort of the potency in a tumor microenvironment. Obviously, there's suppressive factors. There's other issues with how you manufacture cells, for instance, that shorten their half-life and ability to proliferate in vivo. So you know, the approach we've taken is to design sort of a module that can be activated by a small molecule, and this is an inducible co-stimulatory element to boost the survival and proliferative capacity of T cells. And so this is the approach we're taking, which we're using essentially it's on switch or small molecule regulated switch to drive proliferation of CAR T cells and TCRs. And we think that's part of the component. The other, I think, is a very important piece is having your T cell therapy engage the endogenous immune system and sort of facilitate those cells that are in the tumor already, which can be, as we know from checkpoints, antigen specific, right? And so how do you re-stimulate that tumor microenvironment? And one of the things we've been working on quite heavily is trying to get our system or engineer our system to facilitate that by expressing co-stimulatory molecules, releasing cytokines to sort of re-energize the immune system in the context of the tumor microenvironment. I think these might be important things.
Yeah, I think, you know, to add, because we're, you know, in Tmunity, a big focus for us is solid tumors. That's a big part of our portfolio and targets. Aaron's points, I think, are very well taken in terms of, you know, thinking about the co-stimulatory factors, et cetera. A couple of other thoughts. I mean, T-cell biology is exploding at a phenomenal rate now. And as we start to think about the applications of gene editing, be that any form of gene editing, and I think the jury's still out there, looking at, I don't think it's necessarily going to be CRISPR, I don't think it's necessarily going to be TALENs, or it's necessarily going to be zinc fingers. I think all still have a lot of promise there, because while CRISPR is new, the older technologies, technologies are also evolving and keeping up pace. So I think we have to keep a broad open mind. But this whole notion that you can really modulate a T cell now, you can gene edit, and you can actually create a manufactured product. I think that's coming of age now. You're going to see trials emerging this year in that regard. Taking the brakes off on PD-1 or putting them on. Yep. This is going to be very important, but PD-1's one factor, right? There are so many other checkpoints and other things that now we have a better understanding of the biology. You can tweak at the T cell. I think that's going to be very important, because in a tumor microenvironment, one thing we do know for a fact is that cells get fatigued, and they get fatigued in a hematological malignancy. They're even more exhausted in that setting. So T cell exhaustion, I think, and overcoming that is going to be key, as well as more refined approaches to the antibody engineering when it comes to CARs. And then obviously there's an explosion in investment going on right now with neoantigen designs and TCR companies, and really upping the pace of TCR discovery. So I think, it's the holy grail, right, for solid tumor. Are we going to crack it overnight? No. But I think we're at the point now where we can start to make some really meaningful impact in solid tumor environments now. Slow but steady, but I think we'll start to see good data emerging there over the next couple of years.
I think that just to expand the conversation a little bit, so far we've talked primarily about T cells.
Yeah.
And they're natural killer cells, or in the case of my company, they're red cells. And I'll just give you one example where we would try to address solid tumors. You take essentially an allogeneic process where you have a red cell, and you can imagine using genetic engineering, either using a lentivirus during the differentiation or an mRNA. You can express whatever you want on the surface of the cell. So to give you some ideas of some constructs we've made, which seem to work in animals. You can take a cell, you can put anti-PD-L1 on the cell, target it to the tumor. Hopefully it has some checkpoint activity. You can put 4-IBB on the same cell in its natural configuration, which you can't do in any other way. You can't give man 4-IBB because it gets eaten up immediately. And all of a sudden, you're stimulating the immune system, and you're targeted to the cell in that tumor microenvironment. Likewise, another approach of using cellular therapy, we have a red cell where we have anti-CD20 on the surface, and you have trail on the surface, and you just blow up the lymphoma cells. And part of what's going on there is you're getting a cell-to-cell interaction with literally millions of copies interacting with the tumor cell, and you're getting biology that's unexpected. Just like some of the biology with the CARTs was unexpected, the biology of these modified red cells is unexpected. You can't use, you're thinking about, in either case, what an antibody would have done, and assume it's going to happen the same way in a cellular environment, because it's just simply not the case. So I believe this field is going to expand to many other cellular types, and each will find its place depending upon the tumor, the limitations, and the specific makeup of that particular tumor. I wouldn't be surprised if people are growing and modifying platelets at some point, right? Why stop at red blood cells?
I think we share that view that this field is going to expand. There's going to be a lot more experimenting with different cell types. Just to add on to what was said before, our belief is that you're going to need a toolbox, whether it's different cell types you're looking at, or switches that are small molecule driven on switches, or gene editing technologies to knock down the tumor microenvironment, going after dual antigens. All of that, I think, is going to be necessary, and one of the approaches we at Bluebird have taken is we need to invest in building a toolbox that allows us to experiment in that way. And I think we've been pretty good at it to date, but now it needs to translate. And that's kind of where we all are, is trying to figure out how to translate those. But it starts with a target. Without a good target, it doesn't matter how many tools you have in your toolbox, you're not going to be effective. So that's one of the biggest challenges, I think, for the field, is finding really, really good targets that are tractable, that you believe in, I guess and that's a challenge for the field. And I'd be curious to hear what others feel about that here on the panel.
Yeah, it's to me it's remarkable. There's like 20 antigens that everyone uses. That's it. And obviously the aspects of neoantigens is interesting, and you start to think, okay, now you have the whole world of antigens. But from a CAR perspective, there really are very few that are sort of validated, and that's why you see this focus on BCMA and CD19 and a few others. this antigen problem has been sort of at the top of the list for us as a company to solve, and we've been trying to do that in different ways. And one approach I think which is quite interesting to me is really using the natural immune response in an augmented way, really, like we've done for our T-cell add-back and haploid transplants for malignancies. You don't know what you're targeting, but if you're using a sort of enhanced or polyclonal population, then maybe you get around some of these issues and sort of let nature take over if it's well controlled and manipulated. But it's a problem, I think, and there needs to be more effort on trying to identify antigens that have very nice signal-to-noise ratios.
So not to put the four of you on the spot, but I will. Should we expect some solid tumor clinical successes in the next 2, 3, 4 years?
Sure. Absolutely.
Yeah, I mean,
I think most of us are
believe
making pretty big bets, and
Yeah.
we wouldn't make those bets unless we believed there was enough science to indicate that there was a decent chance of success.
I mean, I'll give you one example, right, that springs to mind where there is a validated target, and that's glioblastoma. So glioblastoma, there's a wealth of evidence now that you can create CARs and TCRs, CARs for sure, that traffic, cross the blood-brain barrier, target, activate, T cells get activated, you get the immune synapse reaction occurring, you see cytokines being produced. It will work in certain populations. Now, how well it's going to work is to be determined, but I think the science is mature enough for a tumor type such as that. Whether it's going to work in pancreatic cancer or a larger cancer, to be determined. But I don't think it's a stretch to say that in the next 5 years we should see more results, clinical results, that will be more compelling that this can work.
And that GBM bar is pretty low, right?
Yeah.
Yeah, I think you're going to see a bigger mix of successes and failures. I think you're going to have to fight through some of the failures in solid tumors more than we have in the liquid tumors.
Yeah, and I think the bridge is going to be the work you guys are doing with BCMA targets. Myeloma, that's going to be the bridge, right?
Yep.
It's where we think of it as a semi-solid, semi-liquid tumor, right? So I think that's going to be the next phase, and the data for BCMA is accumulating. It's starting to look really, really compelling. So that I think will be the next natural evolution to step to further understanding for solid tumors.
Any questions from the audience on the solid tumor front? Our third and final topic we wanted to get to in the last half a dozen minutes or so is actually one that I think is near and dear to your heart, David, on the allogeneic off-the-shelf approaches to treating some particular difficult-to-treat diseases. You mentioned earlier in your introductory comments about red blood cell technologies. You want to expand on that?
Yeah, I think there have been now several attempts, and some companies are making progress in different types of allogeneic approaches. And the only reason people are looking at allogeneic approaches is because they don't want to have the complexity of the one patient, one therapy, and the supply chain and everything that goes with it. The challenge becomes there's other trade-offs. So it just has some examples, and I'm not an expert here, but you can imagine if you have an allogeneic T cell, then you have to knock out genes so you don't have graft versus host disease. You don't know if that particular product's going to last as long as perhaps a more natural CAR. It's hard to know, and they'll have their own set of manufacturing issues. Natural killer cells, as I said, which are sort of allogeneic, right, if I understand correctly, in and of themselves. And then what I'm most excited about is these red blood cells. It's not just for cancer, and I know this is really an immuno-oncology meeting, but just to give you another example of what's possible, we've taken red cells and we put catalytic enzymes inside the red cells. So you can imagine putting in an enzyme, for example, to break down asparagine and starve the tumor. You can put in an enzyme to break down phenylalanine and treat a disease called PKU, where there's really nothing that works today. You can put uricase inside and you can treat acute gouty arthritis. So the point I'm trying to make is that cellular therapy, while what we're doing for liquid tumors, and particularly CD19 tumors, is very exciting and important, it's just the beginning of what cellular therapy is going to be, and it's not just in cancer, it's going to be across multiple fields.
I think this year, the next 2 years, hopefully, you'll see more clinical data emerging. I mean, we saw a glimpse of that last year, right, with the data from Selectis, early days, right, 2-3 patients. there's still this issue of graft versus host disease that everybody's struggling with in the background, right? And I think, that if you look at the way the curves have shifted in terms of gene editing capabilities and the ability to now pack so much into a cell, you've got to believe that this is doable, right? But conditioning, all the other things that go into play in the autologous field equally apply to allogeneic patient management, right? So next 2-3 years, if that initial body of evidence starts to really accumulate from companies like Selectis, I think we've got good traction for us to evolve to that. But I still believe that's something for the next decade. I think autologous will be here and now for this, for the next 5 years. But I think with the advances that have happened on the gene editing front, absolutely allogeneic therapies can become a reality as long as we can really come to grips with the graft versus host disease challenges.
I mean, so to give you an idea for the Rubius, the red cell therapies, we'll be in man at the end of 18. You'll see clinical data end of 19 or in 20. And the manufacturing issues you all spoke about, which is very important for a CAR-T, we're going to make this stuff, and in fact, we are making it in stir tanks. It's closer to making an antibody. So all of a sudden, you're making hundreds or thousands of doses at a time. So now, of course, we have to prove that it clinically works. So it does a great job in mice.
It's interesting that you said those timelines, and they're real timelines now, because you look at 2012, when the first proof of concepts occurred in 2-3 patients to sort of catalyze the CD19 field. 5 years later, we're looking at regulatory filings and approvals. So there's something here, right, where the speed of science and progress in the clinical, as long as the clinical efficacy holds out and the benefits risk ratios maintained, we don't see, hopefully not horrendous, SAE issues. I think, you're going to see a wealth of data emerge, and I think this is a great time for investors to really double down in these early technologies, because I think by the end of the decade, we're going to see some hopefully super results for patients.
The other thing we didn't talk about in detail, I don't want to spend a lot of time on it, in the more classical immuno-oncology world, which is essentially antibodies, or sometimes small molecules, but we're going to start seeing all these combination trials read out of PD-1, anti-PD-1, anti-PD-L1, in combination with LAG-3, TIM-3, the OX40s, I mean, the list is tremendous now, right? And some of that can be done maybe by a T cell that's modified. Some will be used in combination. Some will be used in some kind of sequence. So you can imagine we're going to be keeping the oncology, hematology field pretty busy with lots of clinical trials.
Yeah, that's for sure.
Yep.
Any last thoughts or comments? Start at your end, Jeff, and come this way.
As it relates to
free
allo
reign.
or just
Free
general?
reign. You can pontificate as you like in the last couple minutes.
So I go back to my comment, I think, before, that it is an incredible, I think, privilege to be a part of this emerging field. And there are lots of bumps and challenges in front of us, but I think all of us, and I know I can speak for Bluebird, believe that all the things we're talking about are going to happen, including allogeneic, even if that is a ways down the road, as you were alluding to us. So it's an incredible privilege to be a part of this, and I just look forward to seeing these next launches and see how they do on the stumble part, to see how we get through these and learn and continue to learn. It's going to be a cycle of learning that we're going to go through over the next decade that are really going to drive not only new innovation, but also the impact that these things can have on patients, which I think are going to be dramatic. It's so much fun to think about the evolution of the field.
Aaron, anything from Bellicum?
Yeah, I mean, I would echo those exact comments, that the innovation part of this field is phenomenal already, and it's going to continue to grow as money flows into it. The allogeneic part of it, I think, is a really interesting problem. But there's other technology out there that might be even more compelling, such as in vivo transduction of T cells. And we've seen some studies now, preclinical studies, mind you, that demonstrate that this at least could work on the surface. Then, really then by creating a true, therapy in a bottle that you might be able to administer. Obviously, there's safety concerns and efficiency issues there, but these type of approaches might also really move the bar in terms of getting these therapies out into more patients.
That's great. David?
So I would just say it's a credit to all the people on the stage here for doing what they're doing. I think they are driven by both the interesting science and the benefit it's going to bring patients. It's very easy to give up. It's easy to see a clinical trial that goes wrong, or a patient that unfortunately gets hurt. And then people think the field is dead. This field is anything but. It's just the opposite, but it will have its ups and downs. And then the small commercial for Rubius, the next investable idea, I promise you, is a little vial about this big with 10 mLs of super blood in it. And it's allogeneic, it's off the shelf, and there you go. You inject it, and you have a cell product that's doing the job, killing cancer cells.
You'll bring that to us next year at this meeting?
I will be in the clinic and tell you what trials we've set up.
Exactly.
Oz, anything you'd like to end with?
Yeah, I think be prepared for some amazing moments for patients this next 2 years. I think the world is going to change for certain families and certain patients. And be prepared for setbacks as well. This is still a very young field.
Drug development.
But I think the speed with which you've seen progress in the past 5 years has been a renaissance for the sector. I think amazing things are happening and anything's possible.
I'd like to thank the panel. We're just ending on time here, so thank you very much.
Pleasure.